Well, good morning. After a long day on Sunday, when we spoke from the very morning up to the night, now we are supposed to combine everything into 15 minutes. So to do so, we got an inspiration in a recent molecular biology paper, very important, by the way. With about 10 co-authors, and in between was, let's say, the Nobel Prize winner Jennifer Doudna and people like that. And so we got inspired by the cyborg metaphor. The Borg. Oh, sorry. The cyborg is a human. Originally, the cyborg is a human upgraded by devices that aid its survival in hostile, that is, non-physiological conditions without imposing on the organisms the burden of additional time or energy-consuming activities. My generation remembers the term just because of science fiction novels, very nice sometimes. The authors had in mind many cybernetic contraptions enabling survival during a space travel. You can broaden it, of course. If you fall into the Labrador Sea, your survival time is 5 minutes. But if you have good contraptions, you can float there like a seal and read Heidegger during all the time. Yeah. So the expression had quickly acquired a more general meaning and came to denote any self-regulating human-machine couple. So if I really would push it, driving a car is also a human, a cyborgian invention. No, but no, there is a Star Trek. So they took the Borg. They are a cyborgs linked in the hive mind called the collective. The Borg co-opt the technology and knowledge to other species, to the collective, through the process of assimilation. One could thus say that they have a diffuse brain that has at its disposal the shared memory of all thoughts, wishes, defense strategies, and so on of their fellows in the universe. And that's why we took this parable. And the biological metaphor came from this paper I mentioned already. Borgs are extra chromosomal genetic elements that can travel horizontally between members of communities and provide them with the necessary genetic survival kits. Now, everybody in this lecture room, of course, knows the horizontal gene transfer. But what they found out it's a very special, very huge genetic elements traveling in communities of bacteria and archaea that enable some of the members of the community become ultra small. 250 nanometers of the diameter of the cell is a lower limit of being cellular. And thanks to these elements, the whole community became a Borg because they could really exchange this knowledge. No, we took this paper and applied the cyborg parable not to the DNA, because the DNA is not alive, but to genuinely living inhabitants of such communities, and ultimately, on the most general level, also to the whole biosphere. Now, take this is a classical monastery. The monastery lives in a closure. The rules inside the monastery differ from the rules around in the countryside. And the gates are very well kept to allow what is to be allowed to go in and out. And this is a parable of a living cell. Whatever you have in a living cell is a tight closure with a very tight control of internal milieu compared to the outside space. But no, if you think it better, there is another closure. This ad hoc closure, we call here closure 2. They are ensembles of cells that sometimes very ad hoc, sometimes long-lived, that can be in mutual, even in mutual contrasts. But as in human life, you can be a fan of the football club. You can be a Christian. You can be a communist, and you can be a father of family, and all these functions in one person, not simultaneously maybe, and yet functioning. So we are a member of many collectives and you belong to all these. So take, let's say, a meadow or a forest. How many functions a single tree has? We are back to the Umwelt idea. And now, so evolution on our opinion, goes into two ways. Singularity of closure 1, perpetuated without any interruptions since the last universal common ancestor. It never ceased to exist. The closure 1 is something that can be just deleted and you die. The multitude of closures 2 is this analogy, affinities, alliances, and so on and so on. They can change in evolution very quickly because of interpretation of what is in and what is out.
Okay. I will stay here. I prefer this microphone.
Okay.
And yeah, thanks, Anton. And my task here was I tried to categorize the functions of this first closure Anton was speaking about. And when in our model, we actually believe that this first closure, represents the boundary between the living and non-living. And of course, the first function, not surprisingly, will be protection from the external environment, right? Because you have this membrane with phospholipid heads which attract water, and inside of the membrane there are hydrophobic tail which do not attract it. That's not very, that's of course quite natural. But what Anton already said about singularity, this closure actually maintains the organizational, structural, and informational continuity of life in evolution. Yeah, it has been the same since the beginning of life, and it is inherited epigenetically. You always need a template for it. And all living cells share the same closure. And of course, it enables preferential transit and energy compartmentalization. So, with the emergence of closure, there started the possibility to self-organize itself towards order, you know. Actually, it enabled the autocatalytic closure in Kaufmannian sense. And therefore, I think we may say that this closure actually enabled agency. Because one of the many definitions of agent, which I found, is that agents are organisms having the capacity to originate causal effects from within their boundaries or from within their self-reproducing and autopoietic architectures. And of course, this autopoietic organization happens, you know, through this closure, and the closure enables the emergence of semi-closed, self-integrated, organized system, which is the first agent, we believe. And thus it enables autopoietic organization, sensu moreno or motio. And with the closure, there is a possibility, to constantly, the cell constantly compares the outside and the inside of these two environments, and compares the outside with its memory structures, which I was also talking about on Sundays, but it's not only epigenetics in eukaryote, it's also inheritance of organelles or horizontal gene transfer, and as Anton mentioned. And now there is like the most possibly most discutable, like controversial maybe argument is that with this separation of two environments, there was a difference made, yeah? And we can say different, which makes a difference, right? And as Robert nicely reminded me yesterday, the word information actually means the formation of the inwardness, right? Or Anton prefers innerness. So is closure a necessary condition for interpretation? We believe that it is. That with cell, there start agency and semiosis. And, well, such phenomenon can you observe on various levels of organization, from single cell to bacterial colonies, or maybe even to human faces. These interfaces can be co-opted, right? And now Anton will finish with some examples of these second order closures, right? Because the agreement
So I just will read it. Due to the trends, contingencies, the vagaries of evolution, living beings and/or their lineages inevitably stray into states of dependency upon the services of others. As a matter of fact, there could hardly exist a species whose members could prosper in mutual independence, axenically, at least not in the long run. There are some examples, but they are really trivial. And so finally, the whole biosphere is one society as Borgs. We can communicate horizontally. So from the very beginning, every living being keeps its closure one. And during the evolution, all the lineages and members of the lineages, communities, take on abilities to communicate with the others, just because the rules, the codes acquired from the very beginning of life overlap all the time, at least to a small extent. So you can very well see it in your own experience when not speaking a foreign language, and you must somehow overlap your questions in a foreign environment. That's it.
Thank you very much for being on time, and there are several questions. Kalevi, please.
Just a little comment relating to my concept of difference. So whether just a closure is already difference, what makes a difference, I would say this we should analyze precisely here, because it may be is that this is difference due in the relation to survival. If so, this is rather from observer's point of view and not from the point of view of cell itself. So therefore, I just want to point out that it requires further discussion.
I don't think so. Simply the emergence of life on this planet was a singularity, and we cannot say how it happened. Before was chemistry. Afterwards, there was a possibility to interpret world and life with codes, with closure, with possibility to communicate with the others. It was not before. So we don't take the statement that life is just some prolongation of chemical processes. Yes, we can. Of course, we can study chemical processes in the cell and learn very much about them. Yeah, I did it when I was sitting in the lab, but it's not. So the difference that makes the difference is emergence of life, of the closure one, and because the LUCA, the last universal common ancestor, was not a single cell, but a biosphere of different cells, the closure 2 automatically came up with that.
Theodore.
I find it hard to put this comment into a question, so it will just be a comment. There is a tension in your talk that is also already in the autopoiesis paper by Maturana and Varela. Namely, on the one side, you have organization, but on the other side, you have topological closure. And I don't really understand this. So I don't understand it with Maturana and Varela. Why don't you stick to organizational closure with topology, lipids and so on, only coming later? Because I think in this would lead then to a good starting point for further theory.
Yeah, but we do, of course. We even showed on Sunday how structures are perpetuated for millions of years without changing the genetic composition of the organisms. So there is this structural continuity all the time.
Yes, absolutely. But I think it's secondary to the organization, to the dialectics and the movement of the functions of whatever in the cell, that the lipids and so on and the structures are necessary to keep the organization going. But it's more important to have the organization.
But they are part of that organization. How can you take them apart?
Let's fight about that later.
But I did not emphasize maybe enough that it's of course not only the membrane. You have like a lot of transducers, transporters, ion channels. That wasn't emphasized, and also cytoskeleton plays a huge role, you know, in these compartmentalization events.
Another question from Alin, or?
That's a quick one, sorry. So this might be a naive question, but the ability to maintain homeostasis, so what you call basically the first closure, is like one of the classical elements of the definition of life. So is what you're saying in the presentation basically sort of arguing against Deacon's last paper, that there can be no semiosis before the cell because you need the homeostasis? And if so, what exactly is the main reason for that, relating to his point that you can actually see similar phenomena between biomolecules?
If I understand well, that you ask me how the semiosis came to an existence, yeah?
No, I'm just asking if your main point is that there is no semiotic process before the cell, and how does it relate to Deacon's last year's paper in Biosemiotics, in which he was arguing like the exact opposite, that you can see some semiotic processes in biomolecules before the cell.
Yeah, but really the origins of the cells I take as a singularity. Okay. Before the chemical processes before can be all this, of course there are speculations, but there are many models, they all can be described but by normal scientific processes of physics and chemistry, dissipative structures and so on and so on. But the emergence of codes is really a business of the cell innerness. And you cannot derive the codes from chemistry. And this is really a breaking point in the beginning of life evolution.
So the answer is you and Deacon start from different dogma, like a different central...
Well, I am a biologist.
Okay. Yeah, I was kind of afraid of this question. But yeah, that's our model is like this in our book, so you can check it.
Thank you very much.